There is enough iron in your blood right now to forge a single 3-inch nail. Not symbolically, actually enough. Measured, weighed, real. It has been collecting inside you since before you were born. One recycled cell at a time, and your body has never once thrown any of it away. Somewhere in your liver, your spleen, your bone marrow, that iron is being sorted, stored, and reused today, right now, while you watch this video. Doctors call it one of the most tightly guarded elements in human biology, guarded so carefully that almost nothing escapes once it's inside you. For most of your life, that guarding looks like nothing more than quiet, invisible maintenance. No symptoms, no warning signs. Just a slow, careful accumulation running in the background of every day you've ever lived. What you're about to learn is why keeping something that carefully, for that long, can eventually turn into the exact problem nobody saw coming. In 1747, a German chemist named Andreas Marggraf burned dried blood down to ash, ran a magnet through the residue, and pulled out tiny black filings. Well, that's iron. The blood was carrying metal. That discovery didn't answer the real question. It just opened it. 2 centuries of research later, we know why. Iron sits at the center of hemoglobin, the molecule that grabs oxygen in your lungs and carries it to every living cell in your body. Without it, that oxygen has nowhere to attach, and your cells suffocate quietly one by one. An adult body holds around 4 grams of iron. That's not much by weight, less than a small paperclip's worth of raw metal. But it's arranged with a precision no factory could match. Roughly 70% locked inside red blood cells, the rest stored in the liver, the spleen, and the bone marrow, waiting to be called back into service. Here's the part almost nobody thinks about. Every day your body loses less than 1 milligram of iron, a trace gone through skin, sweat, and small internal wear. And every day, it needs about 20 times that much just to keep building new red blood cells. The math only works because of recycling. Old red blood cells, worn out after about 4 months of service, are broken down by specialized cells in your spleen, and their iron is pulled out, cleaned, and sent right back into circulation. Almost none of it is ever wasted. That recycling runs through 2 specialized proteins. A transferrin, which ferries loose iron through your bloodstream like a delivery truck. And ferritin, which stores the surplus inside your liver like a locked warehouse. Together, they mean almost no iron ever needs to come from a fresh meal. Your body is, in a very real sense, running on the same iron you were born with, just endlessly reused. And there's one detail in all of this that barely gets mentioned in textbooks, unlike almost every other nutrient your body handles. Iron has no active exits. Your kidneys can flush out excess salt. Your liver can break down extra sugar. But irons, the body can only slow down how much it lets in. It has never developed a way to actively push extra iron back out. For most of the 20th century, biologists believed they had already mapped out how a cell dies. There was apoptosis, a clean programmed shutdown like a cell folding itself away on command. There was necrosis, messy, traumatic, the result of injury. Two categories. Two answers. As every textbook agreed. There had been strange exceptions along the way. Cases where cells died in the presence of certain compounds without fitting cleanly into either category, but for years. Those cases were treated as lab noise. Oddities to be explained later, not questioned now. Then, in 2012, a research team at Columbia University led by Scott Dixon and Brent Stockwell ran an experiment that didn't fit either category. They exposed cells to a compound that killed them, but the cells didn't shrink and fold like apoptosis. They didn't rupture like necrosis. Something else was happening, and it traced back to one single element. Specifically, iron sitting inside the cell was reacting with fats in the cell membrane, triggering a chain reaction called lipid peroxidation. The membrane itself catching fire in a chemical sense from the inside out. To prove it, the team needed more than a hunch. They measured a byproduct of the reaction, a molecule called malondialdehyde, a telltale sign that fats inside the membrane were being chemically shredded. The signature was unmistakable, and it was everywhere the cells were dying. The researchers gave the process a name, ferroptosis. Iron-dependent deaths. A third category nobody had written into the textbook. This should not have been possible. Iron is supposed to be one of the safest, most essential elements in the human body. The same element sitting quietly in your blood, carrying oxygen, doing exactly what it's supposed to do. And yet, under the wrong conditions, that same iron was acting as the trigger for a cell's own destruction. One of the compounds behind this, a molecule called elastin, had actually been discovered nearly a decade earlier. In a screening study looking for chemicals that killed cancer cells carrying a mutated gene called RAS. Nobody at the time understood why it worked. Oh, it just did. It would take almost 10 years and a proper name for anyone to explain what elastin had been doing all along. The paper was met with the kind of silence that follows something too big to immediately process. Some reviewers were unconvinced. A cell death pathway that had gone unnamed for the entire history of modern biology seemed to many scientists more likely to be a measurement error than a real discovery. But inside laboratories, something had shifted permanently. If iron could do this inside a single cell in a controlled experiment, then somewhere in the human body, under the right or wrong conditions, it was probably already happening. Nobody had proof of that yet. Not in 2012. That would take almost a decade to arrive. Right now, inside you, this exact chemistry is sitting one step away from happening. Every cell in your body contains a small, carefully measured amount of iron, held just far enough from the fats in its own membrane to stay safe. Most of the time, that distance holds. In an adult body, that balance is being maintained inside roughly 200 billion new red blood cells your bone marrow produces today alone. Each one built with its own carefully measured portion of iron. Each one trusted not to let that iron drift somewhere it shouldn't. Your cells go on living, dividing, doing their work, and the iron just sits there, useful, contained, unremarkable. You will never feel that balance directly. There's no sensation for it, no warning light. The only sign it's still working is the complete absence of anything happening at all. But that containment isn't a law of physics. It's a balance. And like every balance your body maintains without you noticing, your blood sugar, your body temperature, your blood pressure, it can tip. It usually doesn't. But it can. Once scientists knew ferroptosis existed, they started looking for it everywhere, and they found it in places nobody expected. In the brains. In a small structure called the substantia nigra. The exact region where Parkinson's disease does its damage. Autopsies showed unusually high iron concentrations in exactly the neurons that had died. If those neurons died from membrane damage triggered by iron, then Parkinson's wasn't just a dopamine problem. It was, at least partly, an iron problem. Researchers began testing a drug called deferiprone, designed to pull excess iron back out of those neurons before the damage could spread. Early results were promising enough to justify a large clinical trial. The final results, published years later, were more complicated. The drug didn't slow the disease the way anyone had hoped, and in some patients, symptoms even progressed slightly faster. The lesson wasn't that the iron theory was wrong. It was that knowing the mechanism and safely controlling it inside a living human brain turned out to be two very different problems. In the heart, during a heart attack, blood flow stops, then suddenly returns. That return is supposed to save tissue. Instead, researchers found the returning oxygen reacts violently with iron already sitting in the damaged cells. And a wave of ferroptosis follows the rescue itself. The treatment was hurting exactly what it was trying to save. In animal studies, a lab-made compound called ferristatin 1, built for the sole purpose of blocking this exact reaction, reduced the damage significantly when given at the moment blood flow returned. It has never been approved for use in a human patient. But it proved the mechanism was real, and more importantly, that it might be stoppable. In the kidneys, the same pattern. In the liver, the same patterns. Different organs, same mechanism, same element. Then cancer research found something stranger still, certain tumor cells. The ones that resist chemotherapy. The ones that come back after treatments. It turned out to be unusually good at blocking ferroptosis. They had, in effect, learned to seal off their own iron from ever reaching the membrane. The same process that could kill a healthy neuron was being deliberately shut down by cancer cells trying to survive. The cells were doing this through a single enzyme called GPX4, whose entire job is to diffuse the exact chemical reaction ferroptosis depends on. Block that one enzyme, and a cancer cell's own defense collapses from the inside. Which meant the reverse was also true. If you could force ferroptosis back on inside a tumor cell, you might kill the cancer the body couldn't. One enzyme, one reaction. One element doing this in the brain, the heart, the kidneys, and inside the cells medicine has spent a century trying to kill. By the early 2020s, more than 3,000 scientific papers had been published on ferroptosis. Several biotechnology companies had moved GPX4 blocking compounds into the first phase of human clinical trials. Not yet proven, not yet approved. But moving. None of it existed in 2011. In 2016, Brent Stockwell stood in front of a room of researchers and said something that would have sounded absurd a decade earlier, that iron. It's the same element quietly folded into your hemoglobin right now. Might be one of the most important discoveries in the biology of disease of the century. Nobody in that room mentioned the paperclip. Nobody mentioned the nails. They were too busy trying to explain how something so essential could also be, under the wrong circumstance, a switch. Go back to that detail from earlier, the one about excretion. Your kidneys can flush salt. Your liver can break down sugar. But iron has no exits. The body can only slow down how much it absorbs. And at the time that sounded like a footnote, a quirk of physiology. It wasn't. That single design gap is the reason ferroptosis became dangerous in the first place. As every other overload your body faces, it has a release valve for. To too much water, you urinate it out. To too much heat, you sweat it off. To too much sugar, insulin clears it. But iron just accumulates, quietly, permanently. In people with a genetic condition called hereditary hemochromatosis, the body absorbs slightly too much iron from food every single day for an entire lifetime, with no mechanism to correct the surplus. By middle age, some patients carry double or triple the iron load of a healthy adult, sitting in their liver, their heart, their pancreas with nowhere to go. That's not a rare edge case. It's one of the most common inherited conditions in people of Northern European descent. As many as 1 in 200 carry a version of the gene that leads to overload. It's common enough that doctors have a nickname for it, the Celtic curse. For how concentrated it runs in populations of Irish and Scottish descent, and it's treatable. In a strangely old-fashioned way, regularly drawing blood, the same way you donate it. It is often enough to keep the overload in check. Well, the hard part was never treating it. So the hard part was noticing it before the damage was already done. And here is what nobody connected until ferroptosis was named. An organ slowly overloaded with iron, for decades with no exit, is an organ sitting exactly one trigger away from the exact chemistry Dixon and Stockwell found in 2012. The hoarding was never just a curiosity about how your body works. It was quietly building the conditions for its own failure, one that medicine didn't even have a name for until a little over a decade ago. It's the same trait that let your body carefully gather enough iron for a 3-inch nail, without ever wasting a measurable amount. It is the trait that leaves it with nowhere to put that iron when something finally goes wrong. There is still enough iron in your blood right now to forge that same 3-inch nail. Your body has spent every day of your life collecting it, one recycled red blood cell at a time, refusing to waste a single fragment. It never learned how to let any of it go. For most of your life, that will not matter. Your body will keep the balance it has always kept, quietly, without you noticing. It has done this every day since the day you were born, without a single day off, without ever asking whether it was worth the effort. But somewhere in a lab right now, researchers are trying to design a drug that can reach into a single cell and decide, on command, whether that iron becomes the thing that saves you or the thing that ends you. If the element your body has trusted the most for its entire existence turns out to have no off switch, what else quietly doesn't?